So, Lasse, Barry, thank you guys so much for being here. It's awesome to finally bring both of you back into the studio. One of the things that I think people always wonder is kind of who are the scientists behind Nucleus? And so I wanted to really feature both of you guys today and talk a little bit about how we all met and also obviously about what we do at Nucleus, because the science is the foundation of what we do, right? That informs everything, that informs what the customer sees, touches, experience, and the ultimate choices that the customer makes. So maybe I'll start with Lasse, our chief scientist. Do you want to maybe give a brief background on kind of where you grew up, where you're from, and kind of how you just got into genetics?
No, be happy to, Kian. Thank you. I grew up in Denmark, in Copenhagen, where I'm born. Then I grew up academically in Sweden, in Karolinska Institute, where I did my PhD. I worked with genetics already back then. I felt it was very interesting, and it happened to be right around the time when the first genome-wide association studies, they came out, 2009.
Wait, what did you get your PhD in what year and what school?
Karolinska Institute, and that was in 2012.
And it was a PhD
in cardiovascular genetics.
Okay.
Doctor of Medicine, cardiovascular genetics is what it says. And I fit right into that. Like, I really enjoyed that kind of like consortium work, get a lot of people together, get a lot of samples, make huge studies, find out stuff. But the one thing I always felt like that I cared a lot more about, that I didn't feel a whole lot of other researchers doing that. So it's kind of like, yeah, well, how do we use it, right? I mean, all fine with that Nature Genetics paper, but how do we use it? And then I started dabbling into these kind of do-it-yourself algorithms and people starting getting their own DNA data. This was at the beginning of 23andMe came up and ancestry.com and those. So people actually happen to have that data themselves. And this was in the beginning meant as a hobby project for me and my family doing that. But then, wow, people liked it. I mean, feel like, oh, can you calculate that? Take my data. And then that became Impute Me. And
So just so people have some context here. So basically, you were doing GWASs, you have a PhD in cardiovascular genetics, and the thing that was really, really pushing you forward was, this is all great, this research is great, but yeah, can we make it useful for people?
Exactly. Like this kind of like application thing is like, I mean, don't get me wrong, I love academia. Publishing is so important, sharing of information. But use it for something that's very near and dear to my heart.
So Barry, how about you? Tell us about your story. Tell us about your education. Tell us about how you ended up here.
Let's see. So I went to college and got my bachelor's in biochemistry.
What did you want to be when you grew up?
Either a doctor or a garbage man.
Okay.
So I went the doctor route.
Okay, got it.
Got my degree in biochemistry, then got my PhD in 93.
In biochemistry?
In biochemistry again. And then did a postdoc at UCSF for a few years. And then...
What did you do in the postdoc?
Worked on transcription. So genes, how genes work to do what genes do, essentially. They're transcribed. So I worked on that transcription. And then I worked at some smaller biotech companies for a few years. And I ended up at Stanford as their director of outreach activities. And in the Department of Genetics at Stanford.
So you were basically the person at Stanford in charge of all their genetics communication. Is that correct?
Yes, that's true. Yeah.
So what did you do there? What did you build?
So we worked with the Tech Museum, which is a science museum in San Jose, where we kind of created this three-pronged program. One was we helped create these exhibits so people could better understand genetics. The one I'm really proud of is we did this thing called the wet lab. And that's where people could actually add DNA to bacteria so that they would glow green. And so we had 6-year-olds doing these kind of bioengineering kinds of experiments. So it was kind of fun. Second part of it was training young scientists to explain science to the public by working at the Tech Museum, and through the third part, which was an Ask a Geneticist section, where people could ask questions. As part of that...
You were then the geneticist?
Yes, I was the geneticist. And then I helped edit the graduate students who answered the questions. And then as part of that, that was part of a website that included this very cool eye color predictor, which we didn't have access to people's DNA, but we could, from what their parents' eye colors were, their grandparents' eye color, we could begin to tell them what their kid's eye color would be.
Wow.
And that was hugely popular. Millions of hits a year from all over the world.
Wow, so it sounds like the through line between you and Lasse is taking deep research that you guys each respectively did and saying, how can we make this useful for people?
Yes.
Right? So maybe it's no surprise that each of you now are here today. And
Exactly.
That's awesome. Well, we have the right people in the room to answer this question. That's for sure. I'll say at the highest level that there's two main kinds of analyses we do. The first is looking for kind of rare genetic markers, things that people can imagine almost in the movies, mutations, if you will, things that really disrupt a gene that can dramatically increase your risk of a condition. One of the most famous examples is like BRCA1, right? Angelina Jolie very famously had this breast cancer marker. So that's one kind of analysis we do. If someone doesn't have one of those rare markers, we then run something called a polygenic score, which Lasse alluded to. And so since we have two scientists here, let me shut up and let me say, okay, well, Barry, why don't you talk about the rare variant analysis we do first, and then Lasse, you can talk about the polygenic scores. Barry, you want to start?
Yeah, so the rare variant analysis, as Kian said, we look for differences in people's DNA that can have a profound impact on their health. BRCA is a good example of one. There are plenty of others. We look at over 800 genes right now, where we search tens of thousands, hundreds of thousands of known variants that can affect people's health. And not only that, we can also look for variants that haven't been found yet. We can actually discover new variants that can affect people. And one of the most important things to me is that we've included a list of genes called the ACMG list of genes. These are around nearly 100 genes, where when people have these, it's not that common that people have them, but when they do, there are actions they can take to live longer and healthier lives. The one that I always think of is called Lynch syndrome. So this is something that will give someone colorectal cancer at a very young age. But if you know, you can actually prevent that cancer from happening by taking certain actions. But 90% or more of people who have Lynch syndrome or any of these other ones don't know they have it. So something like Nucleus can really help these people find this out and live longer and healthier lives.
Wow. Yeah, I mean, and we saw recently a patient had actually hemochromatosis, which I believe is on the ACMG list.
It is, yeah.
And I think his name is Ethan. This is public, and Nucleus had effectively helped save his life.
Yes, and solve a mystery. So he was in this kind of brain fog, being tired. There was something wrong, and the doctors could not figure it out. And he took our genetic test, quickly saw that he was positive for hemochromatosis, and now the cure or the treatment is really giving blood once a month.
Amazing.
And that's it.
Amazing.
And now he'll be fine.
So DNA insight to action.
Exactly.
Right? The thing with these rare markers is they're rare, right? Most people don't have them. So historically in genetics, basically that's where genetic analysis would end.
Yep.
But sort of post-Human Genome Project in the last decade or so, there's been a rise of something called polygenic scores. And Lasse has been working on polygenic scores now for 10 plus years probably. So if someone doesn't have one of these bad genetic markers, which most people don't, we then will run this polygenic score. So Lasse, do you want to talk more about polygenic scores? Maybe even break that term down more and give people kind of an understanding of what it is and how it works.
I'd be happy to, yes. And I think you introduced it well, is that they are what takes place when you don't have the rare pathogenic variants. And luckily most of us don't have that. That's kind of implicit in the word, that when you don't have a rare genetic variant, then typically your genetic risk will instead be composed of many variants, and it can be anything from 100 to 1,000,000 little markers around the genome. It can be single nucleotide changes, or it can also be small insertion or deletions, typically common. The polygenic risk score, what it does is that it makes the calculation of combining all of them. Simply put, you take in a new genome from a person, and then you count up all the variants that person have for this specific polygenic risk score. Then you add up the number of risk alleles, so the ones that will increase your risk, and you multiply each of them by the effect size. And all this have been derived from these genome-wide association studies. That results in a score, a number, basically. And in our research and a lot of use of these big public biobanks that exist out there, it allows us to then rephrase it as a sort of risk estimate for you personally, sometimes only based on your genetics, but also very often we try to calculate in other non-genetic factors. Classical example would be, for example, coronary artery disease is also very much affected by your sex and your age, and if you smoke or not, your cholesterol levels, stuff like that. And the goal really is just to have the best predictors that we can have for every single disease that is out there. And sometimes genetics plays a big role, and sometimes it plays a smaller role. And one of the things I'm very proud of is this system we have designed where you first look for the rare variants that Barry talked about, and then if not, then you go into this more probabilistic approach to it. And the reason I'm proud of it is that, well, it's both good communication for the person at hand, but it's just a very good example of unifying what otherwise often is known as the two spools of genetics, the rare variants and the common variants. And really, I don't think it should be two spools. It is something that is ripe for unification because everyone has genetics and it will impact everyone. Our job is just to communicate that in a good way.
And so it's amazing because, to Lasse's point, historically there was this kind of Mendelian analysis or variant analysis or polygenic scores. And Nucleus has now unified, to Lasse's point, these two schools of thought. So we do both. And we do that through the power of the whole genome test. And what that means now is we can do both kinds of these analysis. And Lasse also said this, we can integrate in relevant non-genetic pieces of information, like your age, your sex, your BMI, your cholesterol levels, and more, combined with your common genetic variants or your polygenic scores into then overall risk. And so really, if you look at Nucleus, we're always very interested, especially for common diseases, in understanding the fact that it's not all genetic. And we recognize that. You mentioned genome-wide association studies. So the polygenic models are derived from these studies called GWASs. Want to talk more about, GWASs, how they work, and, where this data comes from?
Yeah, I'd be happy to. So a genome-wide association study is, as the name implies, something where you associate the genome with a phenotype or a disease. And most simply put, I guess you would just say you take a bunch of people who have a disease and compare them to a bunch of people who have not. Typically, you want them to be matched for things like age and sex and ancestry. But really, that's what you do. And then between those two groups, you go base pair by base pair throughout the genome and calculate, is there any variant that is more common, more frequent in one of the groups? And it can be either. It can be both a protective or a risk variant. And once you've done that, you come out with a set of variants, which can be many or a few, and each of them have an effect size, meaning how much difference was there between the diseased population and the healthy population. And that's that's what a genome-wide association study is. But it is the raw material that goes into calculating these polygenic risk scores. And I do want in this talk to really give a big shout out to all the researchers who have done them, have built all the biobanks and databases out there, because we all depend on them. All actors in genetic space does, and they're a huge resource.
I think the important thing for customers to understand is that we rely on the highest powered GWASs. Right? Mm-hmm. Nucleus as a company sits on the shoulders of giants. The research we use, which is cited in every single one of our disease reports and our trait reports, you can actually see the exact paper we use. We even give actually the set of SNPs that we use to calculate the score. It's all actually publicly available for anyone to see. And goes back all the way to the ethos of actually IncubeMe, right? And being very transparent and very public in how we do our science. And it relies on what really, if you look across all the different studies, it's millions of people and thousands of scientists, I would say. So Nucleus is based off of millions of participants and thousands of scientists' work. I don't think that's an exaggeration to say, in synthesizing these results to the consumer. You think that's a fair characterization?
Oh, I'm one of those scientists, but yes, I have a fair number of GWAS studies too, and I've contributed, but of course, everyone have, and that is such a big resource.
Barry, I'm curious for you, as someone who thinks a lot about science communications, what do you think when we present polygenic scores to patients? What are some of the considerations in your mind?
It's a great question. One of the things, getting back to an earlier point that Lasse made that I'm really proud of, is our transparency. That if they want to look up the paper that we use, it's there, the SNPs are there, they can dive into it and do what they want to do. I think that's really important. I think the other really important thing that we do really well with polygenic risk score communication is we show that it is not it's not deterministic. This is something that we will never be able to pinpoint exactly. There will always be error bars.
Because as people know, if you take a disease, Lasse used the example of heart disease, right? Obviously, people intuitively know some of it's genetic, but what you eat, how you act, if you exercise, right, what your cholesterol levels are, these all impact your risk for condition.
Exactly.
So even if you built the perfect genetic model, you still couldn't perfectly predict if someone would or wouldn't get heart disease or any cancer or any common disease, right?
Right. But that's even true if you discount the environmental factors. If you're just looking at genetics, you'll never just pinpoint their genetic risk is X. It'll be between X and Y. And I think that's really important, this nature versus nurture argument. So I'm really proud of that.
Yeah, I agree. So one question we get a lot is, how accurate is Nucleus? The first thing I'll say here is that we're a clear certified and CAP accredited laboratory. So we've shown that our test has 99.9% analytical validity. What that means is basically if we call a genetic marker saying, hey, in this chromosome, at this position, have this variant, we're very confident that you actually have that variant. We have a collaboration with Illumina, the largest sequencing company in the world, actually. And we use their newest machine, the X Plus machine, to process all samples in the United States on US machine. Another thing to think about in terms of accuracy, what people are trying to get at is, okay, if, I have this risk or don't have this risk, how do I know it's correct? And the way of answering that question is by breaking down, once again, into the kind of two schools of genetics. So let's start on the kind of rare variant side of things, right? Barry, how would you talk about the way in which we do and determine the pathogenicity of rare genetic markers and give those out to patients? You want to talk more about that?
Yeah, I'm really proud of our system again, of how we do this. So we look for the rare variants. We analyze specific genes and look for rare variants. We have a set of rules that we use that can help both look for ones that have been previously found and those that are new or are novel. And one of the things that we do too that's really important is we then have a human check in the system. So we have our lab director look and confirm.
Our lab director is a clinical geneticist, right?
Yep.
And so he actually will personally go through all the flagged markers.
Exactly. And I think that's really important. I took a test with another company that I won't name, but they told me I had a serious condition, for a genetic, that Lynch syndrome that I talked about earlier. They actually said I had. And we looked through the data they gave us, and I've had my DNA sequenced 5 or 6 times in my life, and I've never seen that before. And that was a company that used almost pure AI at this point to do this.
In other words,
the human check is so important.
In other words, they used pure AI and gave you back garbage.
Yes. And told me I had a disease that I don't have. That is, if I trusted them, I would be getting colonoscopies every year from when I was 20, but obviously that's been a while. But it was really hard news to get. Important news if true, but really bad if not true. And so that's these false positives, it's called, where they call something that isn't there, we're really set up not to have that happen.
Yeah.
The customer is what matters most to me.
Yeah.
I want them to get the best results possible, live their healthiest lives, and not be scared by the wrong findings.
Absolutely.
Filtering is a very small word to describe something that currently involves, I think, more than 50,000 different filtering rules that are applied to every single person that goes through our system and are constantly being updated and tuned in a dance, basically, with the human reviewers. And the goal basically is to be as high throughput but also accurate as we can in the future.
Wow.
And it's really important, I think, to mention too that it's not static. We update the variant list every 6 months, I think, is our current cadence. As new variants are found, or variants that were thought to be okay are actually pathogenic, or even the other way. I think that's a really important point as well.
New genes as well.
New genes as well, yeah.
You know, we're a whole genome test, and you know the genome has 20,000 genes approximately, and not necessarily all those genes are necessarily well elucidated. But the beauty of once you have your genome in the cloud is as research improves, we can continually update our analyses and give customers more and more insight into their kind of disease risk. So that's the power of sort of a modern day genetics business. So we talked a lot about rare analysis and the accuracy there. Let's now switch gears to polygenic scores and talk about how do we think about our polygenic scores, ensuring the accuracy of our polygenic scores. Do you want to talk more about that?
Yeah, I can launch into that. I think we already covered how complex actually accuracy is. It's many levels. You mentioned how just something simple as measuring the right genotype is actually very, very important for these things. And I think we covered that. But it is important work that goes into that.
And just so people have a good intuition here, obviously the first step of running a genetic test is to make sure that the marker that we call in a specific position of your genome, you actually have that marker, right? Because obviously if you don't have that marker, any subsequent analysis you do on what that marker means will be wrong. Right. So the very first step is ensure that kind of validity of the file, and we're very proud to be a clinical laboratory, and we've shown that our test has 99.9% analytical validity, which means that if you have a marker, we're very, very confident you have that marker. The gold standard of testing in polygenic model is testing in an independent cohort of people, right? Whether it can sufficiently basically stratify disease risk. And so, for example, let's say I do a GWAS in breast cancer, and that means I take a bunch of people with breast cancer, let's say a bunch of women with breast cancer, and then without. I get these weights. I find that certain SNPs are disproportionately associated in the case group or the individuals with the disease. I then say, okay, well, if this model works, I would expect if I take another group of women who have breast cancer and don't, and I basically test the model, I expect the model to basically identify or stratify the women with breast cancer versus those without. And then you find in this independent cohort, which you can almost think of as a customer, as almost the smallest possible independent cohort, right? You find that the models work quite well. Is that a good articulation, you'd say?
I think so. I think it's, we often see that with people reacting to their polygenic risk scores, like, hey, how can you know this is true for me? And the answer is really, you can't know it for a single person. But the good answer then is to say, well, if there were 100 people like you that we had recruited from somewhere else, we would see that distribution of outcomes. And that is something we can verify and test and do all the time, both us, but also other research groups around the world.
And that's when we give people a probability, right? Because it's basically saying, hey, people with your genetics, right, have this % chance. So one way of thinking about this intuitively is, if going back to breast cancer example, let's say, someone asks, what's the chance of getting breast cancer? Someone would say, oh, it's 13%. Right? Meaning, if you looked at the general population, it's 13%. So if you took a completely random sample of all the women in this country, you'd find 13% gets breast cancer lifetime. The reality is, though, some people will get breast cancer, some people won't get breast cancer. In other words, depending on someone's genetics and environment, their actual risk might be up to 50%, 60, or even 70, 80% in the case if you have a BRCA variant, or it might actually be like 5%. And so what the PRS model does, the intuition for people, is it takes these overall risk assessments and basically will stratify for people to tell them what is your unique risk based off your DNA. And it's based off these massive biobank studies, many, many papers published in Nature and very well elucidated and well-tested models that shows that these models can actually stratify risk. And what you actually often find the customer saying is, wow, I actually had a family history of this thing, and this checks out. And obviously, family history is not a perfect measure, as we know in genetics. But what we find is that, wait a second, maybe there's something that's been running in your family a long time, and then actually someone has a very high genetic disposition that would have been missed by the rare variants.
Yeah.
And so this is what's so interesting, because classically genetics was just this kind of rare variant analysis. And someone would say, oh, my mother had breast cancer, my grandmother had breast cancer, but I don't have a BRCA variant.
Right.
And then the best you could tell the patient is your risk is 13%. But actually, it absolutely is not 13%. And that's where we run polygenic scores, and then we can stratify women. I think it's from 5 to 50%. I mean, you can attest to that, right? So that's the power of using both technologies, both schools of thought in one platform, right? And so everything we do at Nucleus is kind of scientific rigor first. That is the foundation, because we know that that is going to potentially change someone's life. And we take that responsibility extremely, extremely seriously.
No, I agree. And I think talking about risk, we want to put in also the environmental factors that we add in to further stratify that risk. So like with coronary artery disease, you get your genetic risk, but then if you're at high cholesterol, you have an even higher risk. But if we just gave genetic risk, they would not realize how at risk they are. And so I think that that's a really important point about the accuracy of our risk assessment as well, that we further stratify.
Do you want to talk more about how we adjust the polygenic models for non-European ancestries to ensure that we can give everyone, no matter where you're from, useful genetic analysis?
I'd love to, yes. I mean, it is a fact that they are more predictive in European ancestry, or at least in the ancestry in which they're trained, which often happens to be European. And I think there's a widespread maybe misconception that that's it and then it doesn't work elsewhere. That is not correct. There is very good studies out there. I've been part of them myself, some of them. But many have shown that this is a quantifiable thing. You can basically say, well, it works here and it works less well here and even less well here. We can quantify it. And that's what we do at Nucleus. So in a word, I would say the answer here is that you give the prediction dependent on the ancestry group that you are. And we call that from your genetics, of course. And then we just basically modulate the variability explained. I know that's a technical way to say it, but it means effectively that our risk estimates will be right. They will be less extreme the farther you go away from the training population, but they will be correct. It will be 20%, and then that means if you take 100 people like you, 20 will have the disease.
Yeah. So basically, Lasse, you're describing how there's a training population used in genome-wide association studies, and when you deviate from that training population, right? Maybe someone's, Southeast Asian, for example, you find that the predictability of the models go down, but they don't go to zero.
They never go to zero, and that's very fundamental to what we do. That yes, it is less predictive, but that shouldn't hold us back. It's still a prediction model, and we're very open about both what it can say, but also what it cannot say. So then we just correct for that fact and give it out anyway. But of course, as always, very well explained.
And you can then still, you find, even if you're non-European, very material stratifications of your risk across common diseases, right?
If the score is already strong, then the modulation obviously matters less because it will still be relatively strong elsewhere. And one thing that is very near and dear to me is also to say that we want to be part of the solution here, right? I mean, it is a problem that they are so ethnocentric, the Q and biobanks, and which is why I very much see us as being part of the solution by basically having customers from all over the world and contributing to better modulation of the risk scores.
I agree. But how do you think about the communications of giving our patients from different ancestries results?
Yeah, that's a great question. Because there's really two schools of thought. There's the school of thought where if it doesn't meet some certain criteria, they don't get a report. And so for some companies, people of African ancestry can get very few reports. I
feel like it's sad and disappointing.
No, I don't think that is appropriate. I think that we've done a really good job of communicating, but implicitly, we don't say outright, that because of your ancestry, this is less predictive. It's more implicit in that you don't really see much of an effect until it's a high or a really low score. And in those cases, there is a big effect, and they should be told that there is that big effect. But in these little effects where we can't predict it, they just come up with a kind of average findings.
And so I think we really, as a company, have spent so much time, so much effort, not just in the science, but in the communications to make sure everyone, no matter where you're from, can use genetics today to get amazing, useful results. And then also, to Lasse's point, be part of the solution, right? Because as a company, we have responsibility to society in building larger, more diverse data sets and ensuring, again, everyone eventually, there's basically no difference in any population because you just get larger and larger biobanks that you use to train these polygenic models. So we're so excited to be leading that future. I want to address some of these limitations about what can we not do as a company. Think of your genome as a puzzle, okay? So the way that we actually read your genome is we basically cut it up into whatever, a million different puzzle pieces, okay? And then we have, when you're doing a puzzle, I'm sure you guys both know this, you have like the complete image, and then you use the complete image and you kind of make the puzzle from there, right? That's basically what kind of sequencing is. You chop up your genome into a bunch of puzzle pieces, you kind of look at this complete image, and you reconstruct it, okay? And what happens is you cut up the genome into around 150 base pairs or so, puzzle pieces, if you will, and then you reconstruct your genome. And what that means is that sometimes there are really, really large genetic markers that are greater than 150 base pairs, right, that we actually miss. They're rare. I think it's kind of a longer tail case where maybe if you're basically a baby that's unfortunately debilitated, that was just born, and there's some sort of, de novo mutation, you'd probably use a long read genome, right, which is almost like going from the car to the Bugatti analogy, right, to get even more out of the genome. But I'm talking a lot here. I want to ask.
I like the puzzle analogy, actually, Kian, like it, because it's not like this de novo is not just from anywhere. Typically, the problem is something has been inserted from somewhere else that destroys it. So if you want to stay in that puzzle analogy, which I really like, it's kind of like, imagine you have that annoying stretch of blue sky, and you have your blue prick, and like where do I put it? I don't know, because it's all blue. And this is the problem with the short read sequencing, right? You have your 150 base pairs, but they're just repeated for this long stretch of genome. Where do you put it? And then that technology does come up a little bit short sometimes. And this is something we're really putting a lot of effort on. Where should we go? Because it is some pretty important diseases that I feel we're not really touching them as well as we should be, but we will be in the future because it is on the horizon. Long read sequencing is something you talked a lot about. I think that's probably in our future.
Yeah. Your question was what else?
Yeah.
So particular variant goes. Short read technology does a great job of it. It won't get everyone's, but it will get most people's. And then we can add on to that long read later.
So sometimes I go to conferences, genetics conferences, and geneticists come to me and say, well, Kian, but here's the list of things Nucleus can't do. And it's fair. All technology have limitations. And whole genome technology in a collaboration with Illumina, it's a short read genome, which has certain implications. Before we dive into those, though, I would say that if you look at what Nucleus is bringing to the market relative to what's existed, it's almost like going from a horse carriage to a car. So we've seen a dramatic improvement in genetic testing technology. And even with any cars, anyone knows there's normal cars and then there's sports cars, right? Of course, those sports cars tend to be more expensive and, almost, I would say, incrementally more useful versus kind of a 10x more useful. That's kind of the way that we think about it. But so that all to be said, there are still things we miss. So Lasse, do you want to talk a little bit more about the limitations of a short read genome, of a whole genome?
Yeah, I can do that. I like the analogy you gave with a puzzle, where it's like a short read sequencing basically is each of the pieces, and I like the New York picture. Let's say that this skyscraper, I know it should be there. This other one should be there. Then one very clear limitation, it is when you have the piece with blue sky, and there's blue sky, and there's a big piece of blue sky, and it's like you don't know where to put it, and that essentially, I think, catches the concept of repetitive regions very well, or gene duplications for that matter. Because there's simply not enough to match it with. And that's a problem not because of the genome, but because of the technology we use. We use this patent short read, and well, it's not enough to catch a large duplication, for example. There's many tricks to get around it. Like you can be lucky and catch the breakpoint of a duplication with your short read. You can do interesting stuff with coverage depth, but by and large, the Bugatti is also on the horizon, and we often talk about it as long reach sequencing.
One of the things that's nearest to Nucleus is not just doing rigorous science, but communicating to the public. Because as both of you know from your extensive work as scientists, scientific communications is so critical. Ultimately, someone needs to understand the results and know what to do with it. Okay? So Barry, I want you to talk more about how do we think about scientific communications and really the prioritization of science at Nucleus.
So this is definitely a subject near and dear to my heart. I mean, Ask a Geneticist was all about helping people understand science. And so we've worked really hard to explain their results to them and to make it as understandable as possible. Lots of imagery that really helps, that have been scientifically shown to help people understand their risk compared to other people. We have all the references there. We're very careful in the language we use so that it's understandable. We try and get it down to the 8th grade reading level to try and help people better understand this really complex subject.
Yeah. So you're saying visuals. You're saying we lead with overall risk versus relative risk. Instead of saying you're at 2x greater risk, we say you go from 5 to 10%, right? People understand that much better. We categorize by different diseases, right? The platform is very easy to use. It's very intuitive. The genetics companies that give out, 80-page PDF files that you have absolutely no idea what to do with it, that's not Nucleus.
No.
Everything's tasteful and beautiful, and it's a true kind of combining, I think, the best of science, scientific communications, and good UI and UX.
No, I agree.
Yeah, right? I mean, I don't really think that's been done anywhere else.
I think the really important point is, too, that if we do fall short, people with a rare variant, one of those things that can really affect them, we give them the free 60-minute session with a genetic counselor who can help them walk through that result so that you get that human interaction again to really understand what those results mean.
I think that's a great point because some of these results are very serious and there's only so much a report can do. You need to talk to someone.
Yep.
And that's the role of genetic counselors. And we give genetic counseling for these patients, a 60-minute session, and the genetic counselor walk through the patient, basically their entire family history, look at the result, explain the result, and explain what the patient can do about the result. Again, one of the hallmarks of our application, just like how we do sort of the computation analysis, and then there's a clinical geneticist. We kind of had the automated report generation, and then we have genetic counseling. Right. We really, really do our absolute best to give people that kind of automated support and efficiency so we can keep costs down and do it for everyone while ensuring the quality where appropriate, having humans in the loop. That is Nucleus.
Yeah, it's a great marriage of the two so that we can reach as many customers as possible and help them and do it as accurately as possible.